let me just, the next speaker is Dalen Richards from the Oregon Health and Science University. And it would talk about a comparison of gene addition therapy in genetically distinct mouse models of classical phenylketonuria.
Thank you. Hi, I'm really honored to be here. My name is Dalen Richards, and I'm a second-year medical student at the Washington State University Elson S. Floyd College of Medicine. I'm excited to share my scholarly project with you that I'm performing out of the Harding Laboratory at Oregon Health and Science University, where I obtained my PhD, looking at the comparison of gene addition therapy in genetically distinct animal models of classical PKU.
You are on mute. You are on mute.
No. There, it said the host muted me. Did you get my title slide? Do I need to start over? Okay, thank you. So PKU is among the most common inborn errors of metabolism, and it's characterized by phenylalanine hydroxylase deficiency. Phenylalanine hydroxylase, or PAH as I'll refer to it as, primarily acts in the liver to metabolize excess dietary phenylalanine into tyrosine through parahydroxylation. And tyrosine can then go down different fates, including being oxidized, being involved with dopamine production, and even melanin synthesis. But in PKU, this step, this enzyme doesn't work properly. And so there are two major consequences to this. One is a decrease in tyrosine, and a decrease in tyrosine means decreased oxidation of this molecule, decreased dopamine production, and hypopigmentation. But more relevant to the pathophysiology of this disorder is the accumulation of phenylalanine in the blood, and particularly the brain, where it can exert its neurotoxic effects. And these include hypomyelination, the inhibition of neurotransmitter production, and neuronal dysfunction that collectively lead to the clinical manifestation in an untreated state of severe intellectual disability, seizures, and a musty odor due to the outpouring of phenylketones from the body, hence the name of this disorder. Thankfully, PKU is on the newborn screening panel in medically developed countries. And so patients are identified right at birth and immediately placed on a phenylalanine restricted diet. Now, this diet is incredibly burdensome on not just the individual, but the entire family unit. And while I'm not going to get into all of the details of that today, it's important to note that access to this diet is difficult in the United States due to the state governed legislature around insurance coverage for medical nutrition. But there's still limitations of this medical diet. For patients that are treated from birth and consistently keep their phenylalanine levels in the target range under 360 micromolar, there's still overwhelming evidence coming out that there are still neurological sequelae of PKU in these populations. And this includes a slightly lower overall intellectual quotient, impaired executive functioning, slower processing speeds, increased prevalence of anxiety and depression. And we can even see in MRI studies abnormalities within the white and gray matter of the brain. Some of the alternative therapies for PKU include cofactor supplementation that's only effective in about 10% of the population. More excitingly and more recently approved is the enzyme substitution therapy, but it has limitations of self-administration of the daily injections, immune reactions, and currently only approved in adults, though pediatric clinical trials are ongoing. But more importantly, and related to the discussion today, is gene therapy, which now includes in the clinical landscape 2 gene addition therapy trials put forth by 2 different companies, BioMarin with their Fearless trial and Homology Medicines with their Phoenix trial. Both of these companies use one of the animal models that we're going to discuss in today's talk for their preclinical research. But before I get into that, I want to talk a little bit about the phenylalanine hydroxylase enzyme itself. This enzyme is translated as a monomer, and these monomers go into the cytoplasm of hepatocytes where they oligomerize together and form a homotetramer in its fully active form. But as in the case of a carrier where there's a wild type allele that expresses a wild type monomer and an allele that, say, let's has a missense mutation that produces a stable monomer that is mutated and not functional, these monomers can still oligomerize and interact together in the different configurations just showed here. And the overall result could could be an overall decrease in the potential enzyme activity. And in fact, this idea was put forward back in 1975 by Seymour Kaufman et al., who did liver biopsies of parents of children that have PKU, so carriers. And what they found was that these carriers only had about 10% enzyme activity, as opposed to what we would expect a carrier to have, around 50%. And even back in 1975, they proposed negative interallelic complementation, a phenomenon of a preponderance of mutant monomers over the fully active monomers. In fact, what we call today the dominant negative effect. And now I'm going to introduce the ENU2 mouse model, which has been the golden child of preclinical research for PKU. It was made with ENU mutagenesis screens and harnesses a missense mutation in exon 7 in the catalytic domain, and they exhibit classical PKU with hyperphenylalaninemia over 1800 micromolar, neurocognitive deficit, seizures, and hypopigmentation that comes handy in the laboratory as we're correcting the disorder. But an important note is that they have abundant, stable, non-functional PAH protein. Now, one of the many studies performed on this animal has shown that correction of about 5 to 10% of the hepatocytes is sufficient to significantly reduce hyperphenylalaninemia in these animals. And much to my surprise, when I was a graduate student and I performed neonatal gene editing in this model and was able to achieve approximately 10% enzyme activity, confirmed with next generation sequencing of the DNA, showing over 10% corrected alleles, but puzzled about the fact that none of my animals were corrected down into the wild type phenylalanine levels, which would be below this blue line here, and only one got into the therapeutic target range under this red line here. So there were a couple of avenues to pursue in terms of the discrepancies of these results, and what we really needed to determine was the location of our gene editing. Not all hepatocytes are treated equal. Zone 1, the periportal hepatocytes, are actually the site of amino acid metabolism and the most conducive environment for phenylalanine hydroxylase activity. And also the number of corrected cells is important. But unfortunately, to answer those questions, we need to use immuno-based techniques. And with the presence of the ENU2 stable mutant monomer, we cannot do any of those assays. So my ultimate conclusion, aside from gene editing being a permanent approach to neonatal gene therapy for PKU, and as opposed to gene addition, which has been discussed today, is the fact that the field needs a new model of classical PKU that has completely absent PAH protein. So that stemmed into my other project as a graduate student, which was the CRISPR-Cas9 mediated generation of a novel knockout mouse. I generated a list of guides that flanked either side of exon 1 of the PAH gene and validated them with a pretty straightforward Saccaro in vitro guided kit. I selected 2 of these guides and handed them over to our transgenic core, and they co-injected it with Cas9. And after screening about a couple hundred progeny, I was able to detect an allele that had completely absent exon 1 and named this allele Dexon 1. We bred it to homozygosity, and the Dexon mouse was born. This mouse exhibits all of the features of classical PKU and has the same phenotype as the ENU2 mice, with the distinguishing feature that they have completely absent PAH protein. We confirmed this with a Western blot using an anti-PAH antibody on the liver homogenants of these animals. And as you can see here in the Dexon 1 mice, there's 0 PAH protein. And this is in contrast with the ENU2 animals that have abundant protein, albeit non-functional. That's detected by this antibody. And we also were able to show this in immunohistology of liver sections, comparing a wild type and knockout animal, where PAH here is in the green fluorescence and it is completely absent in the Dexon 1 liver sections. Next, we wanted to explore whether the dominant negative effect was real. And we did this by comparing heterozygous animals in these 2 animal models. And as you can see here, the Dexon 1 mice show up to 60% wild type activity. And this is in stark contrast with the ENU2 animals that only have about 30% enzyme activity, confirming definitively with in vivo evidence that yes, the dominant negative effect certainly does exist. And this actually has implications for gene therapy. So gene therapy that's aimed at delivering wild type monomers to these animals. So we decided to employ a gene addition therapy on these two cell lines using a liver-tropic AAV8 capsid with a liver-specific promoter driving PAH, similar to the platform being used in clinical trials, and a dose escalation. And you can see here that I haven't quite filled in all of these sample sizes here, but we harvested the animals 2 to 6 weeks after vector administration, and these were the results we got. First of all, the blood phenylalanine, we knew this therapy was going to be very robust in these animals. And you can see in the medium and high dose animals, all of the animals have robust correction of the phenylalanine levels, and both of the low dose animals struggle a bit in that area. But what was more striking to us was the liver PAH enzyme activity. And as you can see here, looking at particularly the medium and high dose treated animals in both cell lines, there's a much stronger, more robust enzyme activity expression in the Dexon 1 animals in comparison to the ENU2 animals. And if you focus on the high levels at 1 × 10 to the 12th vector genomes, and the ENU2 animals, they max out at about 20%, whereas the Dexon 1 animals enzyme activity is pushing 100%. And then the crown jewel of this project was actually showing the immunohistology of these animals. And this top right, the PAH is in green. This top left panel is our high dose animal at 1 × 10 to the 12th. And you can see the decrease in green and localization of the PAH around these pericentral regions, the vasculature, and which makes sense as we delivered this in a systemic route. And this bottom right panel is a negative control. And you can imagine how this would be helpful in optimization of gene therapies before they move into the clinic in terms of wanting to target zone 1 periportal hepatocytes. So some of the conclusions we came to with this study is that yes, in fact, the dominant negative exists in the ENU2 animals. And while it doesn't hamper the robust efficacy of gene addition therapy at the doses and the duration that we studied in, that we looked at in this study, preliminary results suggest there are dampened effects when particularly looking at the enzyme activity, and that this does have implications for the longevity of the therapy. We also were able to conclude that the Dexon 1 animals do not have PH protein expression and therefore don't have this dominant negative effect. As a result, they have more robust responses molecularly to the gene therapy with particular regards to the enzyme activity, and they're also able to be evaluated with immuno-based techniques. And for that, for those reasons, we do conclude that the Dexon 1 model is going to be superior for the development of novel gene and cell therapeutics moving forward. And while the impetus of this project was really to reduce the confounding variables of the ENU2 model, the overarching goal of this work is to expand the experimental toolbox available for PKU scientists around the world. Interestingly, another group just published in Nature another novel knockout model containing a nonsense mutation at codon 7. And while I'm disappointed they didn't cite my work, I think it's really interesting that their model recapitulates what I found in the Dexon 1 animals, although they've yet to do gene therapy in this model. But it is yet another tool to add to our toolbox for PKU research. And for future directions, I'd like to increase the animal numbers in each of the cohorts and perform gene editing in the Dexon 1 animal model, and also disseminate this animal model for PKU scientists around the globe to advance PKU research. With that, I'd like to acknowledge our collaborators in Switzerland, particularly the Tony Lab. Dr. Tony has been a wonderful collaborator with us. I'd like to thank the Harding Laboratory at OHSU, who are like family to me, and I couldn't do this work without you. I'd like to thank my medical school, the Elson S. Floyd College of Medicine, for supporting me in all of these efforts. And finally, the National PKU Alliance for their funding and support. Thank you. I'm happy to take any questions.
Thank you. Very nice talk. So there are 3 questions. They are, of course, they are about the effect, the negative dominance of your that you have demonstrated by in your model. So there is one question. It's very hard to follow the thing. Which mouse model would better resemble patients?
Yeah, that's a wonderful question. And that brings up a really good point. If you want to recapitulate what happens in the human population, over 75% of the alleles in the human population have missense mutations. So in fact, there is a reason to still do gene therapy. In the INU2 model, but I would argue that in terms of optimizing an approach in a model that has less confounding variables, that would be a more ideal situation. But in terms of answering the question directly, the INU2 model with missense mutations is going to more accurately depict the human population.
Yeah, and on the same line of thought, there is a question about the fact that it would be interesting to study the compound heterozygous NU2 Dexon 1 to demonstrate the dominance for the NU2.
Absolutely.
Yeah, that's another thing. And I think most of them are about that. Message them. There is one that I don't understand. There is how did the message level or the messenger level of transgene compare between NU2 and the other model and the Dexon one?
The vector genome copy number? Is
no,
that
I
what the question is?
RNA messenger RNA, I think. That was the question. I don't know if this is yeah.
We didn't look at the messenger RNA level of the transgenes. I believe that was the question. We did not look at that.
Yeah. And then I have a question for me. It's for me. What you're gonna do with for the negative dominance? So is there a new therapeutic avenue?
I think that there certainly is an avenue to pursue here. I mean, there can be modifications potentially to the oligomerization domain to kind of prevent the oligomerization with the missense alleles or the missense mutant monomers as a potential therapeutic option. But I think, right now it just opened the door to this is something that we can now address as we move forward with gene therapeutics into the clinic.
Okay, thank you. I think we should move on, otherwise we'll be very late.
Thanks.
Yeah.
Thanks.